Thank you, everyone, for this a great opportunity in these last few days at the CRCG event. I think it's been a fantastic workshop. We've heard a lot of really good feedback. I'm Darby Kozak. I'm the Deputy Director of the Office of Generic Drugs, and it's my pleasure to moderate this final panel session. As mentioned throughout this workshop, there are many complex scientific and regulatory aspects that need to be considered by both industry and the agencies to facilitate this transition for the low global warming potential propellants. I think what we've heard over and over, and I think what we're going to hear about more today, and I think it's really good to emphasize, a critical part of this transition is communication, communication, communication. I think all parties need involved. This is a great sort of first start to it, but I think what we need to do is to continue that communication beyond today. This is why you hear quite often from FDA highly encouraging everyone to come and talk to us early and often. I'm going to remind everyone that both the new drugs and the Office of Generic Drugs have different ways in which to engage with us. In particular, our meetings to confidentially discuss any current thinking, challenges, proposals, or recommendations for your specific product or transition. I think one thing I took away a bit earlier was the comment made by Dr. Lionberger, where he mentioned that a pre-ANDA meeting can be submitted to discuss a transition for an ANDA now. You don't need to wait until the low global propellant NDA gets submitted before you can even start your discussions and start your engagement with FDA through both sort of channels. In addition to getting the agency's feedback through these meetings, I really want to emphasize the fact that it also gives FDA a better understanding of the challenges that may be needed to be researched and addressed by the agency. I think we heard a bit about that in the previous panel. Industry may be at the sort of ongoing sort of stages, even what they consider mid to late sort of stages in development programs, but that information hasn't necessarily been communicated to the FDA as much and frequently. So these meetings are a great opportunity to have that engagement, help us work together in terms of addressing some of these issues and concerns. So I want to also add my encouragement to that. You should really utilize these opportunities to engage the FDA on this transition. And they provide critical information that helps inform our thinking. And as we've heard most often, it continues to evolve. There's multiple stages. We need to basically understand what we're doing there. So in addition to today's event, I also want to aspects that need to be identified. I think we heard a lot about the uncertainties, and this is what I really want to focus this panel session on, is better understanding, which you'll see from our panelists, and I'll introduce them shortly. We've got a lot of the industry speakers that we heard from before, both from the propellant manufacturers, the new drug manufacturers, and the generics. I think one thing the agency really needs to hear is what are the critical uncertainties still? We may have perceptions within the agency what those uncertainties are, but we really need to understand from industry's perspectives, what are the uncertainties? What are the aspects that we can provide through the regulatory, better clarity and understanding, as well as understanding both back and forth of where we're at. So I think this is going to be the key element that I want to discuss today. I think one thing that I really want to emphasize also is that this dialogue is really going to help advance both the science and this transition process, with that real key aim being patient access to these medications. The more smooth the transition process we can make, the more ensured that we'll have a continued supply for these medications for these patients without any disruptions and things like this, critical medications. So as I mentioned, with that said, thank you for entertaining me on that sort of dialogue, but I think it's really important to emphasize. I want to introduce our panel. You've met them all before in our previous sessions, but we're going to welcome back Stein. He's the Center Director at Inhalation Products from Cadavia Drug Delivery, Dr. Rick Lostritto from Lostritto Consulting, Lucas Silva, a Senior Specialist in Analytical Services Department at Nanopharm as part of Aptar Pharma. Dr. Mark Boelens is the Global Senior Director and Product Steward at Toxicology at Honeywell. Dr. Markus Laubscher is the Head of Business for Energy and Materials Cora. Dr. Poonam Gulati is the Senior Director at GSK. Dr. Uwe Niesner is the Head of Respiratory and Biologics at Versaris. And Dr. Siva Vaithiyalingam is a Senior Vice President and Head of Regulatory Affairs at Cipla. So with that, I'd like to start with our very first question. And this is sort of that retrospective, learning from what we've been and where we can. What do you think are the major challenges and lessons learned from this previous CFC to HFA propellant transition that are applicable to our current low global warming propellant transition? And in particular, are there some distinct challenges or lessons learned from the new drug approvals as compared to the transition that's going to be happening for the generics? So I'll start with this. I'll ask Stephen if you've got some sort of comments in this sort of space, and maybe Rick, if you want to add on after, and then the other panelists, if you guys have sort of additional comments.
I'll say that I think in this transition, I think we're starting ahead of where we were in the last transition in terms of, I think there's a lot more industry understanding of the products than there was. I think things like elastomer changes aren't going to catch us as much by surprise as maybe they did in the first transition. So I think some of those things are ways in which we're ahead of where we were. What was the end of the question? I'm sorry.
I think in terms of not only from the perspective of that, but is there going to be differences for lessons learned from the new drug approvals compared to the transition for generics?
Yeah. Rick, you want to jump on that one, that part of it?
Well, so listening to what Darby was saying, I'm going to frame my answer in terms of uncertainties and risk.
That'd be great.
With the previous change, there was uncertainty and risk across the board. As Stephen mentioned, there were surprises with elastomers, there were surprises with performance, there were surprises across the board. Things have changed. We have a lot less technical uncertainty. We know a lot more about how MDIs behave. We know a lot better how to design them, test them, use them. I think it was said we know more about the disease states that they treat, and so on. Today, well, back then, there was less regulatory uncertainty. New NDA, no other choice. There were clear overall CEDAR guidelines on how to, produce and submit, and so on, an NDA. Situations flipped-flop a little bit. We have less technical uncertainty, but higher regulatory uncertainty. As of now, you don't know how to submit, what to submit, or even when to submit. And as was said, already behind the curve in that regard compared to some other areas in the world. So that's the flip-flop there. Right now I see it's regulatory uncertainty. And I wanted to reframe what was mentioned as risk earlier this morning when Brian gave the summary. Some of the things that were mentioned as high risk were not because they were technically uncertain, but because it was viewed as regulatory risk, that the health authorities may not accept the concept of multiple changes or the concept of, adjusting the valve dimensions and adjusting some dimensions in the actuator to focus in on the critical performance attributes of DDU and APSD, and with quality by design and so forth, a lot of this was just unknown back in the 90s. Today, it's in use, and as was pointed out, going back to 2016, the literature started really leaping ahead with this kind of technical information. But to me, right now, the biggest hurdle is regulatory.
Maybe if I can add to that, I think we all thinking back to the first transition and looking at it and looking at the timescales at which that happened, and we feel that's a reference point. But I think from what I've seen, not only the major drug companies, but also the generics are jumping so aggressively at this transition that in my mind, this will be a much faster transition, and it will have to be a much faster transition because we don't want to have those same drug shortages that we had in the first transition. And I'd love to hear, what the generics say about this.
I do agree with the last two speakers about what is in critical path. For me, even now, for transitioning into a using a new drug application as a vehicle, there seems to be a better clarity based on what I heard yesterday morning. I think the ambiguity, what I'm seeing is on the generic space, maybe for right reasons. I'm so glad Lionberger just jumped in and said, come on board, just give us what you think, what you want to do, what your proposals, we'll listen to you. That's a good start, in my opinion. So I think there are some uncertainties, but I don't think it is insurmountable. The dialogues and with negotiation, we can get through it. Thank you.
And in terms of the propellant suppliers, and I'm representing Cura here, the Zephex brand, which has been around for the last 30 years. We introduced the HFAs into 134A into the market. And we are serving right now 70% of all MDIs globally, 95% of what's happening in the US and Europe. And we have been very active over the last 10 years to prepare this transition. So that's the other difference. We have seen this coming, we have been preparing, and we have invested a lot of time and effort to make sure that it's going to be smooth. So, you know, from starting with the toxicology studies, from starting to ramp up smaller scale production so that our partners can start to do their trials. And now most recently, actually just last week, we announced our breaking ground of a large scale facility that will serve the market's needs globally on 152A.
Thank you. And maybe kind of getting back to that sort of the uncertainty, is there certain, you know, components within the CFC transition to the HFA that we can leverage to address some of the uncertainty, especially like scientific? I know there's definitely a difference, physical chemical between those two, but is there aspects there that we can actually help to leverage and reduce in terms of the uncertainty, critical properties or understandings within the CFC to HFA transition?
So I actually, you know, lived through that working in the industry at that time. And there was a couple of things happening simultaneously in addition to the CFC to HFA transition. That was a resurgence in interest in leachables and extractables through the other independent events that were happening in the field. And in general, at that point, the MDI space was already aging in terms of how products were developed, tested, and so forth. So you had an aging infrastructure of meter dose inhalers. You had a new propellant change coming along, and you had a heightened new interest independently brought about on leachables and extractables, specifically nitrosamines was the bad actor at that time, and carbon black. And I recall that the elastomers that were used in the conventional, widely used MDI valves used for CFCs were just loaded with carbon black and the PAHs by comparison. So there was a great push on to not only have function, but to have cleaner function. So pre-extraction of elastomers and looking at different materials, getting away from Buna and getting more into EPDM and other more suitable elastomers. But there were always surprises, peroxide residues and so forth. And, you know, we can get into the weeds of specific compounds that I still reverberate in my mind, like tubercaptobenzothiazole and a few others. Some will recall. It was a great effort to get clean materials that functioned well. Then there was the issue of the metals used in the canisters. Some of the formulations were more chemically aggressive, and aluminum canisters became impossible to use for some products. Anodized aluminum could have little minute cracks. You could get perforation of a canister. The valves themselves were under different higher pressure. They had to be redesigned to withstand higher pressure. Different extraction profiles and different materials were being used in an effort to try and have a more robust, you know, stable functioning platform. Certain valves that were in common use couldn't be used because their very structure wouldn't allow them to handle the high pressures. And it went on and on and on. The actuators needed to be redesigned. It was an early part where they started looking at individual tools, cavities in a tool to see how they were similar or not. So the entire industry was being retooled at that point to what I would consider the current modern era. So stop there.
Yeah, some of the canister coatings that were utilized in products after the CFC transition are helpful for some of the issues that we'll have to deal with the new propellants. In terms of, so that reduces uncertainty, but as Geraldine said, many of these coatings, you know, if you go down the supply chain, eventually you're looking at PFAS related uncertainty. But you win some, you lose some.
And maybe kind of one of my draws of my questions with that sort of maybe lessons learned is we hear things about like the critical attributes in vitro sort of properties that may be important and how we link those back to the clinical output. Do you think there's aspects there? You know, I think we've talked about APSD and DDU being important compared to some of the other properties, and always being the question of, well, what's the clinical relevance? What's the you know, that sort of thing. Is there things within the CFC to HFA transition that we may be able to better leverage or understand? Or is this sort of so significantly different that maybe they are too disjointed to be able to link together?
Well, we certainly saw changes in plume geometry, spray pattern, unavoidably because of the different working pressure and just the basic chemical nature of the materials and the way they expand and flow. So yes, we did have to deal with that in the industry, and we had to deal with it again later at the agency when we started to see these products come in, and we had to figure out what was relevant and what was expectations as far as consistency for things like spray pattern. In terms of DDU and APSD, I think some of this more or less applies the same, very similar today. There were some questions with assay sensitivity. So how many actuations constituted an acceptable capture, whether that reflected a dose or one actuation or multiple actuations, were subject of great concern at that time during that transition. It's since been ironed out, of course. Particle size, stage groupings, these were all things. Let's remember the cascade impactor, as was used then, was really an air sampling device meant for monitoring pollutants in the atmosphere. It wasn't designed for measuring fine particle mass in a medical aerosol. So it was an adaptation of a technology that was already, to use a term, bulky and difficult to understand. And it's only later when flow dynamics were better understood and next generation cascade impactors came along, and it was better understood what was happening in terms of particle size and airflow that these began to get ironed out. And I think today we have a much better handle on that.
One last thing I'd say, anybody who I think went to RDD probably saw Chet Leach show a slide looking at the radio label data deposition for QVAR versus CFC beclomethasone dipropionate. So, you know, there was PK study showing significant difference there. So just the increase in understanding of the influence of the output aerosol on the interaction with the patient, I think we certainly have learned from since then.
Okay, thank you. I'm going to go to the next question then. The next question we want to better discuss is, what from industry do you guys still see as the greatest scientific challenges for developing these local marine potential propellant MDIs? Maybe from like an excipient perspective, we can have Mark speak a little bit about like from that.
Sure. Yeah, I think from a material compatibility perspective and also a manufacturing perspective, the properties, the safety profile of the excipient propellant, obviously, is something to not to be neglected, because it has consequences with regards to safety and flammability concerns, etc. So that is one. And then from a preclinical and data generation perspective, you've seen the long list with tox studies, some of them acute and others high tier chronic. What we have seen over the past decade is a consolidation of CROs, and big pharma today still has a lot of weight getting prioritized, but for a small excipient producer like us, we are having difficulties because the options become limited due to the consolidation. So if I would name a challenge, that's definitely one of them.
Yeah, and I would like to highly commend AstraZeneca and Honeywell for the risk they're taking with HFO. I mean, 10 years ago, we went through a list of about 400 compounds in order to find out which ones would we think would be the most suitable to go forward. And we were looking at our experience of toxicology, of manufacturability, of costs, of all the compounds. And we ended up with 152A because ZE, has the unsaturates. It has a lot of impurities that are unsaturates. It's a cardiac synthesizer. So there's a lot of challenges there on the tox side. And I'm really happy, that you managed to get through the tox and so that we have the option to have other propellants out there. But, there is still a lot of things that we need to better understand.
So the question is on the scientific challenges, right?
Yes.
So I think on matching the critical quality attributes, like it could be APST, could be DDU, spray pattern, plume geometry. I feel getting those primary indicators straightened out, which becomes the enabler for conducting PK study, be study with the PK endpoint, or a clinical study. I think this particular pathway that we discussed yesterday is going to be the most challenging because we have to match it. It is as good as developing a generic product. The number of times we heard the PSG yesterday from Office of New Drugs, that could be the number one word that we heard. Product specific guidance, right? So matching those attributes, it's going to be difficult. You need to tweak the formulation. You have to see the stability. You have to see the interactions. You have to see how impurities are being generated, stability, and what kind of modification need to happen in the device, right? This is going to be challenging. The next challenge would be if you're using a propellant, how you're going to store it, house it, how you're going to use it for manufacturing. Do you need a different line than the current line you have? So I think there are a couple of challenges. I think there should be good dialogue also about what difference is acceptable, right? We can't box ourselves to the existing statistics that we use for deriving the equivalency. So we have to think, is there more relevance? So that still we are equal. So there's going to be a criteria should be redefined. What is good enough to say, okay, this is comparable. Let's go from in vitro to PK study. Okay, this is comparable. Let's allow them to go for a streamlined clinical study. So I think that is also going to be a challenge. Thank you.
So I want to just follow up on that. I think you hit something important on the head and I want to add to it. Matching the critical performance is one thing, but what I see as a challenge is a thought process whereby, and I'm going to disagree a little bit with Geraldine, where you make the absolute minimum number of changes possible. And I disagree with that. You make the right number of changes that make your optimal performance right along what you want. So I think we have to get over the concept that if you change a little bit in the valve and you change a little bit in the actuator, that risk is additive. And if you change a little bit more in the coating or something else, that's also added. You keep adding the risk. No, the risk is not meeting the in vitro targets that was just mentioned. So I think there needs to be the risk is in where to start and how much to change. I see, as I mentioned in my talk, I see all these as button levers and dials that you can adjust to get the target delivered dose and APSD profile as close as you can, and to use that to your full advantage. So that's the point I wanted to make to add on to that.
I think from a formulation perspective, for example, at least from my experience, what we've seen, I think the available excipients like ethanol, I think that's definitely something you can use to try to modulate to match the performance. Or as well as we've seen like modulating the valve volume, that's also something else that you could use. I'll probably say don't over-engineer it. I think I'll probably go keep it simple. And again, just trying to match the delivered dose and APSD. And probably as an advantage, probably if you look back at the CFC to HFA transition, I think nowadays there's a little bit more, let's say, automation as well to remove the variability during the testing. And I think that's actually something our advantage.
Yeah, no, I just wanted to add, Rick, I think the regulators, according to my experience, are receptive. If good justification, it's not overburdened with changes, but the right changes and the matching the profile in vitro, in vivo, and actually what the patient gets, that is the prime important, I think, for regulators as well as for the technical people here in this group.
It was just mentioned about the consistency of the automated test method that you talked about, and it was part of your presentation, and we chatted a bit about that. I think this is really important. I know from direct experience when I worked at BI that little innocuous changes to the test method, such as the interval between firing or the position in which you shake, some of these can have a dramatic effect, particularly in development where you're still honing in on what you want to have. What I would hate to see is that in the zeal to exactly duplicate what you have the incorrect shaking procedure for your new product, or the incorrect interval between shaking, or the incorrect priming duration, or the incorrect number of shots for priming, that then kicks you out of everything else simply because of this little change upstream. So when you optimize, you have to optimize everything all over again. It doesn't mean those are big changes. It means you're paying attention to what your new product is, how it's functioning. And the testing regime is very important.
Feel like I feel pretty confident with both propellants that they're technically viable and efficacious products to be made. I feel good about that. I do think the option 1 bar is still a pretty high bar. And, you know, I'm encouraged that it does seem like there's some flexibility. You know, my company lived through the development of a generic Symbicort, and I think I counted like 45 different metrics we had to show bioequivalence on. And hitting 45 for 45 is not an easy thing. So I'm encouraged by some flexibility. I will say I understand the rationale for multiple PK studies for each product strength with the charcoal block on certain ones. But I will say even with the product that we had, what I thought was a really, really good APSD delivered dose match, I was terrified waiting for the PK results. And so I think that's still a big risk and uncertainty.
And to follow up on that, maybe, and this kind of leads into the next question too a little bit, is I think what I hear is there's a lot of components where we can develop a lot of feedback in terms of what we need as inputs and outputs. How do we, in terms of engaging with FDA, to have those communications, to have this? Because I think when we see these applications quite often, it's already we've done everything we think is going to be expected and everything's successful. Where at the point do you think is good in which to, or there are avenues already to have these earlier sort of discussions on, this is the background, why we're developing this way. These are the variables that we're tuning to that. And maybe I do it from both perspectives, from the new drug, we feel that the new drugs get and then the generic drug, like avenues in which to communicate the FDA at these early sort of stages with that. Or is there need or feedback, what we need to do to have those sort of communications?
I can speak for the generics companies from the regulatory point of view. So yeah, it's I think known and we are experienced professionals and know when not only to share the good data, but also the justification if something does not work. And then particularly if you think that some of the plans do not work, seek agency advice in advance, make sure you're on the right track still, and then course correct if needed. So definitely encourage not only to share the very good data, but the real and scientifically justified data that's coming out.
Yeah, I agree with that wholeheartedly. However, I see a reticence on the part of some folks in industry to reach out because they're afraid they're going to get, slapped on the hand or they have to have specific questions. But I think if FDA remains open and receptive, as they indicated they would be, I think that's going to be very useful. Venues like this are very helpful too. Certainly, it's been, I think, extremely useful conference. So thank you for doing that.
Thanks. And I think one thing there is that we do want to, like I said, is seeing that sort of data early, understanding the justification early definitely helps frame what is feasible, what are challenges, what are opportunities, as well as like still the uncertainties. Do you mean? I think that it's beneficial on all parts if we can have that conversation. I will get now to my next question. This is probably one of the things there is, where do you still see the greatest regulatory uncertainty or challenges for this local development MDIs? And describe what you think and where areas of clarity would be most beneficial and or sort of aspects that would be for the business opportunities. And we heard a bit earlier, preventing the abandonment or not even the starting up of a program. What are the uncertainties that would be beneficial to both the new drug and as well as the generic drug industry from a regulatory perspective?
So
I can, oh, go ahead.
I
Go
can
ahead.
make a start on that. Thanks. So I mean, essentially, you know, I think the key challenges from a regulatory perspective are the uncertainties in what are the data requirements and the extent of the data requirements. So in terms of going out to health authorities, even having that early engagement to seek scientific advice has obviously been very beneficial. But then you encounter the next level of challenge, which is, you know, very different, often very different feedback from different health authorities. And then it's the, you know, up to the sponsor then to navigate these differences that you're hearing, because ultimately you need to decide on what is going to be your program of clinical studies, for example, and your more or less clinical really, to identify a clinical program that will give you, you know, essentially a pathway for approval into each of the territories. So I think that's the key challenge that I've seen, certainly from a regulatory perspective.
Yeah, I would completely agree. It's we are used as global pharmaceutical companies that there are different regional requirements. But after the RDD conference earlier this year, it looked like for the very same change, one of the most divergent regulatory requirements from European Union, possibly a CMC variation with the CMC package only, up to FDA, possibly a CE study and a full ANDA development and an ANDA submission. But I see here already movement before this conference and a lot this conference with Sally Seymour and Li Chang, that there is acceptance that what first looks like the streamlined approach may in the end, if you just literally look at all the requirements that you would need to fulfill, it's actually not a streamlined approach, option one even. For the CE study, possibly, but you have additional realistic APSD assays, dissolution assays you have to develop. You need to do this charcoal and non-charcoal block PK studies that need to be prepared with preliminary studies. You have Q1, Q2 differences. So you would need to develop possibly computational models in order to justify that, and the bar to meet all the in vitro requirements and then on top the in vivo requirements that look like a very big burden, but I see movement here and a request from the industry and both generics and pharmaceutical, the originator industry, that to see science-based, look which parameters are most relevant and not check the box of to fulfill all and then you move on, but really by that mean that it's a science-based tailored approach to greatly reduce the regulatory risk. And with reducing the regulatory risk, we heard from Geraldine, you reduce the overall risk to the program and internally may be able to really justify viability of the program on a global view.
Yeah.
Just the last thing, which is additional for the generic industry, it's availability of the RLD. We heard the request to source RLD at this point of time, but when you're not sure what kind of steps the RLD is doing in the future, and you need RLD at the right age, at the right time, at the right site. And the PK studies, particularly the pivotal PK study, maybe a few years ahead. So the uncertainty of RLD, and there are ways to mitigate that from the company perspective as well as from the regulator perspective, but these will take time. And if that occurs in the middle of your development program, that may impact the development program.
Yeah, I want to echo that. I mean, also from the part of the propellant producers, there's really a plea to the regulatory authorities to be more pragmatic, to accelerate this transition, because ultimately what will drive this transition is economics. As Geraldine said, and I can't say it better than she did, it is the economics of the bigger gas market of refrigerants that is going to drive this transition much faster than anybody's thinking, because the existing propellants, if you want it or not, they're going to be very expensive very quickly. You see it with 227EA, which has gone through this a bit earlier. With 134A, it will be the same. So if we're not ready with the new propellants quickly, we will get into these problems of having too expensive propellants, which lead to too expensive products, which leads to discontinuation of certain brands. So a very strong plea to see that we can make this transition happen quickly, because technically we have everything we need, and we have done a lot of the testing already. So I think it's really about the interdependence that is in this room of all the people who can make it happen, that we hold hands and we actually go through this.
I also want to add my voice to that. You're not alone. There's a lot of people that think that way. It's not a matter of if, it's a matter of when, of what you exactly said will exactly happen. The time for action is here. What I saw today, looking at the regulators from around the globe, is that there is an apparent need, and I'm going to share this thought as we discussed with Richard Darby earlier. There's a need for harmonization between the regulatory health authorities, obviously. And we were musing, is there a possibility for something like a mini ICH effort? We know ICH takes a long time, but something like it, where the regulatory parties come together and try to agree on some harmonized criteria for in vitro and maybe in vivo. Because it's not just this country, it's not just Europe, it's a global economy, global manufacturing system. And most of these companies are multinational, global, and they have to have separate dossiers, separate applications, separate methods, for different regions. It adds to the cost. And it also adds to the uncertainty of whether they should continue or drop out as well. So there is a call for that. And I don't know, I've heard some talk, I think Markham mentioned there is there's a group of inter-regional regulators that meet and discuss this. And I saw some slides up about that. I would urge that effort to continue and try and achieve some results.
So we did have a few more, but I know we've come at time, and I appreciate the panel session here. I think one aspect that I do want to definitely say is understanding what are the critical aspects that are, the certainties that are needed, communicating those, as well as in terms of your development program, business sort of aspects that help inform like what you're going to do starting your development program. Having that sort of context, I think from my perspective and maybe from the agency perspective, helps understand like the bigger sort of picture, as well as timelines and priorities from what the agency and how industry can work and communicate better in this sort of transition process. So with that, I am going to ask Dr. Robert Lionberger to come up. He is the Director of the Office of Research and Standards in OGD to give some closing remarks. And thank you for our panel.
So thank you. So all you on the panel have to stay here and listen to me. So first of all, I want to thank everyone who participated in this meeting. I want to thank our CRGC colleagues and collaborators for doing all the logistics and planning and organizing. I want to thank our OGD organizing committee, especially Brian, for his work on getting this all together. I really appreciate Sally Seymour and all of our colleagues from the new drug side who came to this. This is very unusual to have this sort of generic and brand common audience. Most times the issues are separated in time. Now they've come together. And so really appreciate all of the effort and the participants of people who helped organize this, get speakers. This, one feature of our CRGC meetings is it's not just FDA staff talking, it's also industry perspectives and learning and understanding on that end. Really appreciate that. So I only have 3 things I want to say at the end. So first of all, I think sometimes with all of this, PSGs are so hard. I would say I've been at the FDA a little over 20 years. Almost all of those 20 years, I've been working on trying to get rid of the clinical endpoint studies for generic bioequivalence for inhalation products. And we're now sort of inches away from doing that. Hopefully within, a very short time, we'll have that option available. And I think that's incredibly significant for this transition. It's sort of thing, PK and in vitro approaches that touches on the harmonization that's much closer to a globally harmonized approach. I think it's the foundation you see when you meet with OND and abbreviated approaches and saying for that. And we've been working on, on the regulatory aspects, trying to coordinate our, within FDA, our scientific research outside to build the foundation for it. And I think that's an incredibly important part of this, and I want to make sure that that's not forgotten. But as we finish that milestone, we're not going to stop working. Then it becomes to making those in vitro and PK approaches more efficient, right? How many PK studies do you need? What's the right way to interpret the in vitro studies? And I think as we get a little bit of experience with approving a new propellant, right? How big of a change is a new propellant? Does it fit into that abbreviated approach very clearly for generics? And I think with the, once you see a few submissions and actual data with that, that'll be much clearer. So in, a several year future, I think you'll see very clearly these abbreviated approaches. Also, we'll know what the right things to look at in the PK studies and the in vitro studies to bridge these clearly once we've seen a few applications of that. So I'm very confident that we'll have much more efficient PK and in vitro approaches in the future. And I do hear the comment, well, you have to do all these tests. What if one's a little bit different? Let me tell you this, what I think the secret sauce on that is. That's thinking about it from either a model-informed drug development, quantitative medicine approach, also physics-based modeling, CFD modeling. Those sort of computational approaches can really be the insight into, does this small difference matter in where things get deposited in the lungs? And so I encourage companies who are really active and want to be in the front to have that capability somewhere in your infrastructure so you can say, oh, look, I noticed a small difference. How do I figure out whether it's important or not? I think those quantitative medicine approaches are the thing that will really help make these approaches more effective, right? Because you're doing something significant. You're approving all these products without clinical data. That's a big step. But that's an important step in the advances of quantitative medicine. A few weeks ago, I was at the ACOP meeting. This is Pharmacometric Society, thousands of people there working on new drug development. I was there on the panel with our O&D director as well, talking about this. And the key challenge and the key value thing is, what are clinical studies that you don't need, but there's a high bar for having the data that supports that. But I think that's an area. So I don't want to make sure people realize the significant effort that we've gotten to the place where abbreviated approaches are on the table across generics and the brand development products. So that's the first point. Second point for the generic industry, as I mentioned in the comments earlier, don't give up on your development programs for the current products. We're worried about drug shortages. There's going to be markets that need those products, but also from a practical standpoint, if you have an approved ANDA, you are in a much better place to navigate any transition in the future than if you don't have one. If you have that approved product, you might have the possibility of supplementing it. But you could also say, well, I'm going to, take this product and compare bioequivalence, submit a new application, but I've got a product and all the data, all the development work done, and my product's bioequivalent to the new RLD. Even if it's a separate NDA, I can make that bridge, but I have all the product development work. You're in a much better situation to navigate the transition if you have an approved product. So I urge you not to sort of get out of the current development programs. But if you are concerned about the transition point, please, and this is my third part, please come and meet with us in the Office of Generic Drugs about your ANDA development programs. We know that for the generic side of the industry, right, these new propellants, they're not in an FDA-approved product, so they're novel excipients. So you can't submit an ANDA today. At some point, you will be able to. You can come and meet with us now on the studies. You can be actively involved in developing with the new propellant. You can meet with OGD about the studies and the comparison. You don't have to wait for the brand product to go to the new propellant. If you can show your product with a new propellant is bioequivalent to the current brand product that's on the market, come and meet with us on that approach, or come and meet with us on conventional versus conventional formulations and make sure you have a plan for the transition in the future. We are willing to engage with you on that. And I think that's an important part of keeping the generic pipeline full. So with that, I really, again, thank everyone for attending and appreciate your participation in this. I've learned a lot about the state of the art in this area. I think all of the other FDA staff who are here also would echo that. And that ends our meeting, and I wish you all safe travels home.